Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing IISER Aptitude Test question paper conducted in the year 2019. I will be discussing only the biology section. If you are new to my channel, please do like, share and subscribe. Coming to the first question. Which of the following produces a dikaryotic phase in basidiomycetes? So, a dikaryotic phase is seen when there is plasmogamy. Plasmogamy is fusion of cells but without the fusion of nuclei. The cell will remain diploid, but it has two nuclei. And this dikaryotic phase is seen in basidiomycetes as well as ascomycetes. So the answer here is option B, plasmogamy. In the next one, in the standard nomenclature used for depicting floral formula, which type of flower is represented by K? In brackets they have given 5. K represent calyx. The subunits of calyx are the sepals. So wherever they have written gamopetalous, you can and syncarpus. Syncarpus is for gynoecium. Carpal are units of gynoecium. So syncarpus also not correct. Gamopetalous. Petalous is for corolla. Petal is the subunit of corolla. So that also can be striked out. So the remaining is polysepalous and gamosepalous. Now 5 in brackets, it indicates united or fused sepals. There are 5 sepals and they are united. That is what it shows in brackets. So it is gamosepalous. Answer is option 5. Polysepalous is when the sepals are free. And gamosepalous when the sepals are united. So the answer here is option C. Then which one of the following statement is true? Blood is a connective tissue which does not secrete collagen. That statement itself is correct. Next one. Tendons are a type of dense irregular. They are not irregular, they are regular. Third one. Adipose tissue is an example of fluid connective tissue. Blood is an example of fluid connective tissue, not adipose tissue. Then saliva is secreted by an endocrine gland. It is not endocrine gland, it is exocrine gland. So option A is the correct answer. Then next one. Match the entries in column 1 and column 2. Which one of the following choices is correct? Heme. Now heme, it is a prosthetic group. And it is prosthetic group of many enzymes like catalase, cytochromes of the electron transport chain, nitric oxide synthase, etc. So heme acts as a prosthetic group. So A4. Next one is NAD. NAD is a coenzyme. So B3, A4, B3, A4, B3 in option B. So NAD is a coenzyme and it regulates various metabolic pathways like beta oxidation, glycolysis, etc. Then zinc. Zinc is a cofactor of the enzyme carboxypeptidase. So C1. And niacin is a vitamin. So the answer here is option B, that is A4, B3, C1, D2. Then next one. Which of the following helps in bacterial motility? So centrioles. Centrioles are not found in the prokaryotic system. So that is not correct. Fimbriae. Fimbriae are usually used for attachment. Microtubules again not found in prokaryotes. And flagella. Flagella is the one which helps in bacterial motility. So the answer here is option D. Then next one. The diagram below shows a typical ECG. If the AV node is not functional, which of the following option correctly represent the changes seen in ECG? So what is ECG? ECG is electrocardiogram and is used in tracing the electrical activity that is taking place within the heart. So under normal condition, electric impulse, they travel from the SA node, then to the atrium, from the atrium to the AV node, and then ventricles will start contraction, and this flow of electrical impulse, it will cause the four chambers of the heart to contract and relax in a coordinated fashion. Now if we want to know the functioning of the heart, we have to take an ECG. Then what does this P wave represent in the given figure? So it is the P wave, and it represents depolarization of left and right atrium, and also correspond to atrial contraction. So it is depolarization of left and right atrium and also atrial contraction. Then QRS complex, this QRS, this peak, it corresponds to electrical impulse as it spread through ventricles and indicate ventricular depolarization or ventricular contraction. Then next comes the T wave, and the T wave follows the QRS complex and indicates ventricular repolarization. That is what the T wave represents. Now what they have asked, if the AV node is not functional. Now I told you in the beginning that the contraction or the electrical impulse under normal circumstances, it will travel from SA node, then spread to the atrium, from the atrium to the AV node, and from there to the ventricular septum. So if the AV node is not functional, the ventricles will not contract. So ventricles will not contract means out of this portion, that is QRS complex, because it represents ventricular depolarization or ventricular contraction, that will be absent because AV node is not functional. So the answer here is option B, the QRS complex will be absent. Then next one, during light reaction in chloroplast of tobacco plants, where is the proton gradient generated? So if you see the diagram, you can see that there is the pigment system 2, where oxygen evolving complex, where the splitting of water in pigment system 2 lead to the accumulation of protons in the lumen. So lead to the accumulation of protons in the lumen. Then the passage of electrons through the various photosystem also make the protons move from the stroma to the lumen side of the membrane. Then protons are used up from the stroma to convert NADP to NADPH by the action of the enzyme NADP reductase. And these above mentioned steps, that these steps whatever I have told here, that is oxygen evolving complex, passage of electron through various photosystems, then the production of NADPH from NADP by utilizing the protons from the stroma will result in creating what is known as proton gradient across the thylakoid membrane. So where is the proton gradient generated? It is between the stroma and thylakoid lumen. It has nothing to do with intermembrane space of chloroplast as mentioned in A and B, as well as our inner and outer chloroplast membranes. So the answer here is option C, between stroma and thylakoid lumen. Answer is option C. Then before coming to the next question, I will like to explain the contraction of muscles. So first what happens is, the impulse or the impulse will travel along the axon to the axon terminus. That is like this. The impulse, this is the neuron, motor neuron. It will travel, the impulse will travel along the axon to the axon terminal. In the axon terminal, it will result in the opening of the voltage gated calcium channels. Calcium will be taken into the cell. That is, it will enter the axon terminal, the calcium ions, and this will result in the exocytosis of acetylcholine into the synaptic cleft. This is the synaptic cleft, and exocytosis of acetylcholine into the synaptic cleft. It will bind to the acetylcholine receptors on the postsynaptic membrane or the membrane of the muscle, that is this one. It will bind over there, and this is your neuromuscular junction. So the acetylcholine receptors, it will bind over there. This will result in the opening of Na positive ion channel, voltage gated Na positive ion channels, and it will result in the entry of Na positive ions into the muscle cell. Then the action potential formed again will be propagated along the sarcolemma down to the T tubule. This is the T tubule. This action potential triggers the release of Ca2 positive ions from the smooth endoplasmic reticulum. These calcium ions, they will bind to troponin on the actin filament. The troponin will be bound by the Ca2 plus ions, and this will result in exposing the myosin binding site on the actin filament. The myosin binding site will be exposed. The myosin head, it will bind to the myosin binding site on the actin filament, and myosin head, it also has ADP and PI. ADP and PI. It is bound with ADP and PI. Next, what happens is the power stroke. That is, ADP and PI will be released from the myosin head, and this will result in binding of ATP again to the myosin head. That is, once ATP is bound to myosin head, the muscle will start relaxing, and the link between myosin and actin filaments will be detached, and and then what happens is the ATP will be converted to ADP plus PI, and again myosin will be available for binding to the actin filament. This is the mechanism of muscle contraction. So acetylcholine will be released. It will bind to acetylcholine receptors. Na positive gated channel will be open. That will result in sending the action potential to the T tubules. From there, the T tubules, it will trigger the release of Ca2 plus ions from the smooth endoplasmic reticulum. The Ca plus ions will bind to troponin and thereby exposing myosin binding site on the actin filament. The myosin heads with ADP and PI bound will bind to the actin filament, that is to the myosin binding site. Then it will result in power stroke. After that, the myosin head will release ADP plus PI, and it will bind to ATP. Once it is bound, myosin head binds to ATP, it will result in muscle relaxation. Once the muscles are relaxed, ATP will again be converted to ADP plus PI, and the myosin will return to its original form. The Ca2 plus ions, they will be transported back to the lumen of the smooth endoplasmic reticulum. So this is what happens at the neuromuscular junction. Now coming to the question. Which of the following will not be triggered by the release of acetylcholine in the synapse at the neuromuscular junction during muscle contraction? So generation of an action potential in the muscles. Yes. Release of Ca2 plus from sarcoplasmic reticulum. Yes, that also is involved in contraction. Binding of ATP molecules to the myosin head. No, if myosin head is bound with ATP, then muscles are getting relaxed, not contracted. Only if myosin head is bound with ADP plus PI, it will help in muscle contraction. So option C is the answer. They have asked which will not be triggered by the release of acetylcholine in the synapse at the neuromuscular junction. Then shifting of tropomyosin to expose myosin binding sites on actin filaments. That is also happening during muscle contraction. So the answer here is option C. Then listed below are different plant reproductive stages: parthenocarpy, syngamy, apomixis, polyembryony. Which of them can give rise to clonal progeny having same genotype as that of the mother plant? Now parthenocarpy. Parthenocarpy, it is production of seedless fruit. It is the process of producing fruits without the process of pollination and fertilization, and it does not produce any seeds. It is a method of asexual reproduction, and it produces progeny that have the same genotype. It does not produce progeny that have the same genotype as that of the mother. So it does not produce progeny that have the same genotype as that of the mother plant, but it produces genetically identical offsprings. Offsprings will be genetically identical, but they will be different from the genotype of the mother. So parthenocarpy cannot be the option. Then next is syngamy, that is fusion of male and female gametes during fertilization is called syngamy. So that also is not correct. Then comes apomixis, is the formation of seeds without fertilization, and apomixis involves formation of seeds without syngamy. So all seeds are genetically similar to their mother plant. Then next is polyembryony. It is the process of producing two or more embryos from a single seed. So polyembryony and apomixis will give rise to clonal progeny having the same genotype as that of the mother plant. So here is option A, that is 3. The diagram below 4 represents the pedigree of a certain genetic disease. Affected individuals are shaded. What mode of inheritance of the gene responsible for this disease? So this is the pedigree that they have given. They have given in the option it can be X-linked recessive, Y-linked, X-linked dominant and mitochondrial. Now here the female is affected and all the children that are born to this female are affected. In this also the female is affected and all the children are affected. Here also the female is affected, so all the children are affected. Here the male is affected but female is unaffected and children are unaffected. So it shows it is a mitochondrial inheritance because mitochondria is having maternal inheritance. It is inherited from the mother. So if there is a defect in the mitochondrial DNA of the mother, the same defect will be passed on to the next generation. Now you can get it confused between X-linked dominant and mitochondrial. It is not Y-linked and X-linked recessive because X-linked recessive means it should show crisscross inheritance, but it is not showing that. Then Y-linked means all the male child will be affected, but here female children are also affected, so it cannot be Y-linked also. The confusion can arise with X-linked dominant. So I have made a cross for a Y, it is not X-linked dominant. So this is X and Y, which is the male parent which is not infected, and suppose this is the female parent where both the X genes are having the disease, carrying the disease. So these are the gametes produced and all the offsprings are affected. So that condition can be satisfied over here in the first generation. But if you see this generation, that is the second generation here, this one, this offspring will be this genotype X, capital H and X, because one X will be obtained from the father and the other X genome will be obtained from the mother. So mother is having the disease, so naturally the child will be carrying the disease and one X will not be affected. Since this is an X-linked dominant, we are thinking that it is an X-linked dominant, so naturally this individual will be affected. So she is married to a male who is not affected. So this is the cross and you will get affected daughters, affected son, unaffected daughter, unaffected son. But that is not the case over here. You get all the offsprings of this particular female affected. Now if you see this condition where, so it is not the case of X-linked dominant. Now if you see the condition over here, this one, where this is the father who is affected with the dominant trait or X-linked dominant condition, and the female is unaffected. So father is having only one X chromosome, so he will be affected, means naturally he will be affected because he is having only one X chromosome. So XHY and X small h, because female is not affected with the disease. Now what is the progeny you get? You get affected daughter and unaffected son. But here you can see that in the cross it is both the children are unaffected. So this is not a case of X-linked dominant condition. It is purely mitochondrial inheritance. So the answer here is option D, that is mitochondria. The next one. An E. coli whose DNA is fully labeled by N15 is grown in a medium containing N14H4Cl. What will be the percentage of hybrid, that is N14, N15 and light N14, N15 molecules at the end of 80 minutes? Assume that the doubling time of E. coli is 20 minutes. So first E. coli is having N15 DNA, and DNA replication is semi-conservative. So after 20 minutes it will be separated into two N15 strands or bands, and it is grown in N14 medium, means it will start incorporating N14 nucleotides. So it will give rise to hybrid DNA after 20 minutes, that is 100% will be hybrid DNA. Then after 40 minutes you will get 50% hybrid and 50% light. So these are the hybrid DNAs and these are the light DNA. So 50% hybrid and 50% light. After 60 minutes you will get 25% hybrid and 75% light strands, DNA. And after 80 minutes you will get two hybrid bands, that is one is over here and the other is over here. My drawing is a little bit difficult to understand, that's why I have given percentages over here. You can redraw in your book on a fair paper and you will understand it better. So 2 out of 16 into 100, that is after 80 minutes, 2 will be hybrids. So 2 out of the total 16 into 100 will be 12.5% hybrid DNA, and 14 by 16 into 100, that is 87.5% will be light DNA. So they have asked after 80 minutes replication, so it will be option D, 12.5% hybrid and 87.5% light. Then next one. The following plots represent the body size distribution of a fruit fly population. Dashed lines represent ancestral distribution and continuous line represent distribution after a few generations. Okay, the dashed lines, that is these lines, they represent ancestral population. These lines, this is the dashed line. This represent ancestral population, and continuous line, they represent the present generation. If the larger individuals have better survival as well as higher reproductive rates, now the larger individual means the one with more larger body size. So if the larger individuals have better survival as well as higher reproductive rates, which diagram below best represent the expected change in the distribution of body size? X-axis represent body size. So this axis is the body size and this axis is the frequency. So they have asked if the larger individuals have better survival rate as well as higher reproductive rate. Okay, that means the present generation should have higher reproductive rate and better survival. So it should have larger body size than the ancestral population. So if you see the diagram C, you can see that the body size of ancestral population is less and the body size of the present generation is more. So that is having better survival as well as higher reproductive rate. So the answer here is option C. The next one. What are the types of immunity acquired by transfer of antibodies to the fetus via the placenta? So which antibody is transferred to the fetus via placenta? It is IgG. And vaccination of an infant. So that is not the question regarding which antibody, whether it is active or passive immunity. Which one is active, which one is passive. Now active immunity, it results when exposure to disease organism triggers the immune system to produce antibodies to that disease, as in the case of vaccination. And passive immunity is provided when a person is given antibodies. The person is given antibodies to the disease rather than producing them with his or her immune system. So that is passive immunity. So transfer of antibodies to the fetus via the placenta. So here antibodies are directly transferred to the fetus via the placenta. That is the case of passive immunity. The fetus is not producing the antibodies. So it is a case of passive immunity, and vaccination is after getting vaccinated, your body will start responding and it will produce antibodies against the vaccine for acquiring immunity. That is the case of active immunity. So answer is option D. Then next one. Which among the following correctly represent the sequence of events for the normal polymerase chain reaction? So it is denaturation, annealing and extension. Denaturation is when the DNA, double-stranded DNA is denatured. It separates into two strands. Annealing is where your primers start to bind to the target DNA sequence. That is annealing. Extension is this primer sequence, it will be extended and the DNA will be replicated. So answer here is option A. The next one. Loss of biodiversity occurs due to the growth of carrot grass, Parthenium species. This is an example of. So Parthenium species is an invasive alien species. And an invasive alien species is an introduced species to an environment that becomes overpopulated and harms its new environment. That is alien species invasion. So the loss of biodiversity due to the growth of Parthenium species is an example of alien species invasion. Answer is option A. So thank you for watching my channel. Please do like, share and subscribe. I have already uploaded 21, 22, 23 papers also. You can go through my channel playlist for the same. Thank you.